Hello everyone, present here virtually. I am Rutuja here and I welcome you all for today's session, which is titled as Significance of Statistics and Pharmaceutical Research. And presenting this session, we have with us our today's expert, Professor Nalini Kanta Sahu. Professor Nalini Kanta Sahu, working as professor and principal in one of leading college, SRM Modinagar College of Pharmacy, under the ambient of SRMIST deemed to be University, Delhi NCR campus, which is NAAC A++ category 1 by MHRD NIRF ranking 50. He is at present with experience of 15 years in academics, administration, and accreditation, training and research. He completed PhD from Shiksha Anusandhan University, NIRF rank 24th. He completed course in statistical reasoning and algorithms in pharmacovigilance, SRAP, Uppsala Monitoring Center, Sweden. He is pursuing doctoral research under the supervision of Professor Dr. Bui, directors of Artificial Intelligence Laboratory. He has 4 design patents published and 1 granted to his credit, along with 61 research and review article in International Journal of High Repute. He published 2 books of international repute. He attended more than 52 international and national conferences. He is editorial board member of 63 international journals. You can start now, sir.
Okay. Thank you. Thank you. Research circle up. Am I audible? Am I audible?
Yes, sir.
Okay. Thank you. So thank you for giving this opportunity to talk today on significance of statistics in pharmaceutical research. So as you know, before I'm just telling you something about myself, as she has told today about me. Myself, Dr. Nalinikan Sahu. I'm working as principal, Department of Pharmacy, SRM Institute of Science and Technology. It's a deemed to be university, and also I'm pursuing my postdoc under Dr. Bui Thang Hong, Vietnam. This is Ton Doc Thang University, and I'm pursuing my PhD postdoc in pharmaceutical research, especially artificial intelligence based some LCMS works. And I did my PhD in 2014 from Shiksha Anusandhan University from Odisha. It's deemed to be university. And coming to today's talk, that is significance of statistics in pharmaceutical research, means I want to speak about what is the role of statistics in pharmaceutical research. Okay. So research, as you know, what do you mean by research? Research is nothing but we are just doing some innovations based on some previous literature review, previous works, what previous works are being done, and what extension in our work we are going to do. Means what kind of extra work we are going to do, so that we are going to do some innovations in this in your for the next generations, means for the society. What kind of you can say what kind of contributions we are going to do, right? So coming to this pharmaceutical research, as you know, pharmaceutical research, I'll tell you first. I'll give a brief on statistics. What are the different statistics we are using means we are learning, and those statistics how we are implementing, where we are implementing in case of pharmaceutical research. As you know, pharmaceutical research is a major field of concern. As you know, the pandemic not over, still going on. In this so much scarcity of the drugs for the COVID-19, and so many works are being done in your vaccinations innovations. So also, so some of the works being innovated and implemented around our almost all the people have been vaccinated. So whole contribution, whole credit goes to the label of higher authorities who have taken this initiative, starting from the research field. In research field who are working, they are having a great contribution to the society. And when they are implementing this research, when they're doing this research, they are implementing so many statistical methods that we may not be aware of those things. So everywhere there is application of statistics are there, right? Everywhere statistics applications are there. I'll come to that point. Like some example I'm giving in drug development. When we are developing some drugs for the society, that drug which is called your that process is called your drug development process or drug discovery process. And the journey of launching of the drug into the market is being inoculated by starting from the research and development team to the distribution team, is marketing team, right? This is journey. And in between, so many works are being done. And in the research level, let me tell you, so many clinical trials are being done. Then only it is going to be implemented in the market, right? So before means coming to the market, means implementation of your you can say this clinical trials, uh, uh, so many phases are there, like clinical trial phase 1, 2, 3, 4, always. Okay, and generally these clinical trials are being done on either patients, and afterwards if it will pass, then it will be done on your I mean on your healthy volunteers. Then if it will pass, it will be tested on your patients. Okay, and generally the main objective of your clinical trials is to see any toxicity of that particular innovated product. Okay, what you have innovated, is there any toxic effect is there or not? That you are going to find out, right? This is the main objective of your you can say the pharmaceutical research and clinical trials. So you can say in pharmaceutical research, two things are there. One is the drug development, and when drug is going to be developed, when drug is developed, it has to undergo some of the clinical trials practice, right? So those clinical trials practice has to be successful. Then only the drug will be implemented in the market. This is the main thing I want to tell you. For that, how statistics are going to be useful, so that I am going to discuss, right? So today's talk, I'll cover almost fundamentals of measurement, what kind of things we are going to measure here, parametric versus non-parametric tests, okay, descriptive versus inferential statistics, then common test for comparing two or more groups, because whenever you are doing some research in clinical trials, let me tell you, you are selecting so many groups, some healthy groups, some patients, some groups you'll be doing with some treatment, some groups you'll be doing with the standard drug. So many groups will be there, right? So you need to compare the results obtained from all the data whenever you are going to do, right? Various data you'll be getting, and on those data, whatever the results you'll be group the means your you can say the object, what object you are going to select. Then you have to divide into different groups. Then you have to implement your statistics, means you have to implement your experiments, and the experimental results whatever you will get from different groups, you need to compare which group is having the better result, right? And how you will compare? Lakhs of results you'll be getting, so many results you'll be getting, and it is very difficult to compare manually. So that is why we need to take the help of statistics. Okay, So that you are going to do. Then correlation, regression, that also I'm going to discuss. What is that actually? But shortly, not in details. Mainly, I'll focus on the significance, where it is going to be implemented, right? And what we are not going to cover here, most non-parametric tests, measures of agreement, then multivariate analysis, statistics and clinical trials, and anything in depth. Means we're not going to go in depth. Just I'll give you the overall brief, right? What we are going to. So next thing is there, it is coming to our mind, why you should care. Why you should care? What is the need of that, right? So one thing, the answer is, without knowledge of statistics, you are lost. It's on the text, right? So whatever the result you are getting, if you are not going to apply the statistics, sorry, then almost you are going to be said that you are lost. Fine. Coming to next term, that is variables. Okay. So coming to variables, that is independent versus dependent variables. Okay, independent versus dependable variables, levels of measurements, then kinds of statistics. Okay, what kind of statistics we are applying? Then level of measurement means which level we are going to apply, independent versus dependent variables, right? So level of measurement, it is coming to mind. We can say this is a kind of statistics that is appropriate, depends on the way the dependent variable has been measured. Okay, so four levels of measurements are there. One is your categorical, nominal, that is called categorical or nominal. Second one is your ordinal. Third one is your interval, and fourth one is your ratio. Coming to next point, what are statistics? Okay, this is a major concern. As I told you, we're going to study about we're going to know about the significance of statistics in pharmaceutical research. So before that, we should know what is statistics actually, right? What are statistics? Methods for organizing, analyzing, interpreting the numerical data obtained from experiments. So as I said you, whatever the experiments you are going to do, it is not like that you are going to apply in your clinical trials. Any kind of experiment you are doing in your lab also, various results you are getting, repetition of the results, okay, repetition of the experiment. So you will be getting so many data. Then you need to apply this particular statistics. Okay, so statistics is a cumulative of organizing, analyzing, and interpreting numerical data, which is obtained, which is obtained from the experiments, right? Coming to next point, that is descriptive statistics and inferential statistics. Two terms are coming into our mind. One is your descriptive statistics. What does it mean? Organize and summarize the data. So descriptive statistics means nothing but here we are going to organize and summarize the data. In the second stage, that is inferential statistics, used to make an inference on the basis of data about the non-existence of relationship between the independent and dependent variables. I'll come later what are independent and what are dependent variables. So you are going to give an inference between these independent and dependent variables. That is called inferential statistics. Means you are going to establish some correlation between these two terms, right? Coming to next is your kinds of statistics. As I told you, when data are measured at the categorical or ordinal level, non-parametric statistical tests are appropriate. Unfortunately, time prohibits much discussion of this important class of statistics. And when data are interval or ratio, parametric tests are usually the correct choice, depending on the assumptions required by the test. So what test is requiring, depending upon that, you are going to apply whether parametric or non-parametric, right? So parametric generally when we are applying, when data are interval or ratio, means there is some interval, means you are giving some, you are applying some drug right now. After 5 minutes again you are applying. After 10 minutes again you are applying. After 3 minutes, 1 hour again you are applying. So in these kind of interval treatments, you need to apply these particular tests. That is your, you can say the parametric test. Okay. And kinds of study in that, as I said you, it is always possible to downsample interval or ratio data to apply non-parametric. So non-parametric test, when you are applying, it is possible for some down sample interval. Down sample means in the treatment concentration, suppose you are changing, right? And it is sometimes also, as I told you, possible to upsample ordinal or categorical data, right? Coming to decisions about level of measurement require careful consideration when planning the study. So, when we are going to plan the study, we need to give some decisions. How we are going to give the decision? So, decisions about levels of measurement. What kind of level of measurement you are going to do, right? So, because sometimes some dependent variables will be there, some non-dependable variables will be there. Whatever the result you are going to get, that you are going to compare, right? So, kinds of statistics. What kind of statistics are there? One is your descriptive statistics. Second one is your inferential statistics. Okay. So, coming to descriptive statistics, data reduction, mainly summarize data in compact form. So we are going to compact the data, whatever you are going to do. We are minimizing the data, right? So minimum, maximum, mean, standard deviations, range, etc. So mean means suppose five means group you have taken, and you are being asked to find the average weight of the total 5 people. So what you are taking, individual weight you are taking, adding all those weights, then you are taking the average of those by dividing 5. So that average, that means it is representing for all 5. Means as a whole, all the people are having average weight this much. So you are minimizing the data when you are expressing the data, right? So that is what called minimum, maximum, your mean, standard deviation, range. So those things we will discuss in detail, right? Frequency distribution, coming to this point, description of data, how many times it is occurring. Suppose you have repeated the experiment, the result whatever you have got the first time, second time whether you are going to get the same result or not, means repetition of the result is there or not. If repetition of result is there, that means frequency 2 times it is happening. Means 2 times you are going to get the same result. So that frequency of distribution means description of the data versus theoretical distributions, and data can be plotted in various ways to show distribution. So when you are going to show the distribution, there is a lot of way to represent their distributions. Okay, it's a common distribution how I have shown you. And measures of coming to this is a distribution of the data. You can say this is a normal distributions. Okay, normal distribution. In this, what is happening described by mean or variance, about which more later. Generally, the frequency, the distribution, it is shown in terms of your, what is that called? Normal distribution curve. Okay. And coming to measures of central tendency, generally 3 things are there to express your measures of central tendency. One is your mean, median, and mode. Mean means, as I told you, average of the results. Median means what? This is the middle value of the distribution. And mode means how much time the particular result has been occurred, right? How many times the result has been occurred. Suppose I'm telling you an exam in one class, 60 people are there. Out of 60 people, 5 members they got 90%. So that means 90% is repeated by 5 people. That means what is the mode? 5 is the mode. That means 5 is the mode to get 90% result. That means how many times it is occurring. That is called your mode, right? And about your descriptive statistics, as you can see here on example, I have given number of cases, minimum how much, maximum how much, range, what is the range how we are calculating between by taking the difference of maximum value and minimum value. So maximum is your 222.222 and the minimum value is your 192.308. So between them, the difference is known as your range. So what is the difference between these two? 29.915, right? Then arithmetic mean, how we are calculating the mean, means how much time is there, total number of observations divided by the addition of the result divided by the total number of observations, right? Then standard error of arithmetic mean, that is 0.229. One example I have given, arithmetic mean, how much it got, 205.154. Standard deviation, how we are calculating standard deviation, the formula is some specific formula is there. That is root mean square deviations. Okay, standard deviation we are finding out in terms of your RMS value, root mean square deviations, right? Variance, how much it came, 35.4. So this is a result of overall one experiment. Median, you can see here 204.08. Mode is about 200, right? Mode is about your 200, means this 200 result has been achieved many times. That is why the mode is this one, right? And which to use actually, where to use, which to use. The mode is appropriate at any level of measurement. Any level of measurement, we can implement the mode. The median is appropriate with ordinal or interval or ratio data. So we can implement the median in terms of your, what is that called, ordinal, interval or ratio data. Then the mean is appropriate when data are measured at the interval or ratio level. The relationship between measures depends on the frequency of distribution, and when data are not are normally distributed, all values will be equal. Means whenever you are going to get some result and you are going to represent the data normal distribution graph, then you can expect all values will be equal. Okay. Coming to next point, you can see different types of graphs I have shown here. This is the normal distribution, as you can see here. This is the mean median mode. This is a positively skewed distribution. This is low negatively skewed distribution. So three types of curves I have shown you, right? And coming to measures of variability, range already I have told you. This is the difference between the largest value and the smallest value, right? And variance, example I have given here, the formula. Sorry. So in the variance is, sorry.
Hello, sir. Sorry to interrupt you. Your slides are not changing, sir.
Now?
No, sir.
Okay. One more. Just let me share it. Now it is shown.
Yes, sir.
Okay. Now it is moving. Once you check it.
Yes, sir.
Changing.
Yes, sir.
Okay. So measures of variability, as I told you, another one is to express your variance, standard deviations, coefficient of variance. That I don't want to talk much about this one because these are fundamental levels. The standard deviation divided by the sample mean is your coefficient of variance. Percentage coefficient CV. We are telling percentage CV. How we are expressing the standard deviation divided by the sample mean, that is called your coefficient of variance. Confidence intervals express the range in which the true value of a population parameter falls with a high degree of confidence. So confidence interval is nothing but when true value of a population parameter, whenever you are selecting any population parameter, in that population parameter, the true value you need to, it should fall with a high degree of confidence, either usually 95% or 99%. Example, I'm telling you, for the f.f0 data, the previous slide, the mean 205.15, and the 95% CL your 204.70 to 205.60. 99% is your 204.56 to 205.75. So the range is narrow because n is large. If number of observations are large, then the range may be very narrow. So the estimated population mean is good. So population mean also will be good. Population mean, when it will be good? When The range is narrow, and I'm telling you, and large number of data will be there, right? And coming to another inferential statistics in that, methods used to make inferences about relationship between the dependent and independent variables in populations based on a sample observations from that populations, right? Coming to populations versus samples, experiments normally use sample statistics as estimate of population parameters. And population parameters are written with Greek letters, sample statistics with Latin letters. So sample statistics generally it is shown in Latin letters, but population parameters they are shown in the term of your Greek letters. Okay. Then sampling distribution, how we are distributing the samples. So different samples drawn from population will usually have different means. In other words, you can say sampling error causes sample statistics to deviate from population values. Errors is generally greater for smaller values, your smaller samples. Okay, so this is about your sampling distribution, how we are going to distribute standard deviation versus standard error. What is standard deviation? What is standard error? What is standard deviation and what is standard error? Sorry for the interruptions. So in between I got a call, so I have to lift urgent call. So sorry for the interruption. So let me continue with the standard deviations versus standard error. The mean of the sampling distribution equals the population mean. The standard deviation of the sampling distribution, as you know, also called the standard error of the mean. The standard deviation of the sampling distribution. When you are distributing the sample, after that, whatever the mean, I mean standard deviation you are getting, that standard deviation of that particular sampling distribution is called your standard error. And generally we are designating as SE, standard error. The standard error is an index actually. And of a sampling error, and it's an estimate. It's an estimate of how much any sample can be expected to vary from actual population value. Okay, actual population value, with that how much difference should be there. So that is what we are going to discuss here, right? About your standard error. So this is a representation of that, which it is showing whether there is any difference between the population mean and sample mean is there or not. Between population mean and sample mean, is there any difference? If there is any difference, that means some error is there. So that is called standard error, and we are going to represent. And some parameters are there, some statistical limits are there. It should be within that limits, right? So the logic of statistical test: first of all, we need to set one hypothesis, then select appropriate statistical test, what test you are going to implement. Then you have to decide, depending upon the result, whether to accept or to reject the hypothesis. So these are the three basic steps whenever you are going to do some statistical test. One is your define hypothesis. Two types of hypotheses are there: null hypothesis is there and alternative hypothesis is there. I'll come later. Coming to second bit, that is your select appropriate statistical test. What kind of statistical test you are going to implement? Next is you decide whether to accept or reject the hypothesis. Okay, hypothesis testing. Coming to that, if you see here, accepting or rejecting the null hypothesis, as I told you, type 1 and type 2 error. Mainly it is happening because you are checking the difference between these two, between the type 1 and type 2, means between the you can say the uh, error sample mean between the sample mean and you can say your population mean. Whether is there any difference between these two or not? If sample mean and population mean is there will be there some difference, then some error is there. And if there is some error, you may say those error may be of two types, type 1 and type 2, right? Type 1 and type 2. So type 1 error represents that is your likelihood of incorrectly rejecting the null hypothesis, and type 2 error beta is the probability of accepting the null hypothesis when it is actually false, right? These two things are very much important. You need to know all these things, right? As you can see here, decision consequences of statistical decisions. As you can see here, decision null hypothesis accepted, null hypothesis true or false, null hypothesis accepted or rejected. It is correct. That means null hypothesis accepted, null hypothesis is true. And null hypothesis, if it is false, but it is accepted, then type 2 error. Type 2 error is there. Null hypothesis is rejected if type 1 error is there. Type 1 error will be there means that means the null hypothesis will be rejected. And what will be the correct? 1 beta, that power, right? So these are so many things, concept you need to choosing of a statistical test. Okay, what kind of test you are going to choose there? So many tests are there. Like your one is your comparing counts, categorical data, that is chi-square test. Okay, so single sample chi-square test access the probability that the distribution of sample observations has been drawn from a hypothesized population distribution. Second one, your example, does self-control training improve classroom behavior? Self-control training improve class behavior? Teacher rates student behavior? So many questions are coming to our mind. Outcome, the observed frequencies compared to distribution of behavior ratings for entire school, the expected frequencies. So these are coming under the test when you are going to implement this chi-square test. Okay, chi-square test for two samples. Whenever you are implementing for two samples, the contingency table analysis used to test the probability. Contingency table obtained from sample frequencies equally, those would be found if there were no relations between the independent and dependent variables. If there will be no difference between these two variables, that is your independent as well as your dependent variables, then you are going to take the contingency table and you need to compare the data, right? As you can see here, example, you can see here one of the publication was there, D. Casuitel, the author in 2008, they have done swallowing ability, okay, success in total laryngotomics who did or did not have radiotherapy. They have some patients they have gone through radiotherapy, some have not gone through radiotherapy, and those are having some swallowing problem was there. So they have done that particular experiment. So they have taken surgery only, and they have taken surgery plus RT. Okay. And coming to swallowing was significantly better in the group that didn't receive radiotherapy. Significant value, that is your 0.5. Yes. Okay. So coming to that is your next point, comparing group means. Okay, group means choice of test can be dependent on number of groups. So what are the different, you can say, what are the different groups you are going to take, and what kind of test you are going to implement. Okay, one is your t-test. Either you can implement ANOVA, that is analysis of variance, or you can implement this ANOVA also. One way ANOVA is there, two way ANOVA is there. So these are there. F test is there also, right? Then calculating, then you need to calculate at different levels. You are going to calculate at different levels, like your 95% confidence limit, or you are going to implement at 99% confidence limit. Okay, so these three things you need to keep in your mind. Like t-test, one is your whether you are going to implement ANOVA. If you are going to implement ANOVA, whether it is one-way ANOVA or two-way ANOVA. And coming to the confidence limit you are going to set, that is calculating 95% or 99% confidence, because these two levels only we are working. 5% or your 1% we are allowing, right? So generally we are working in these two levels. So coming to this t-test, student t-test, it is called your student t-test. And in this student t-test, student with example I am telling you, what is the main objective? Compares the sample value to a hypothesized exact value. One value we are hypothesizing, and with that hypothesized exact value, we need to compare the sample value. Suppose example I am giving you, students with hyperactivity receive self-control training and are assessed on a measure of behavior. The experimenter hypothesized that their average score after training will be exactly equal to the average score for all the students in the school. So what the mentor will do? He will take the average score of the training, and then he will take the average score of all students in the school. Right? Then he will compare both the data. If there will be any difference, then he need to report. Right? So as I said, you the sample mean as well as the population mean. So two independent sample t-test. So that is in this what is happening? Test hypothesis. That means of two independent groups are not equal. So those independent groups are not equal. Means those groups between that he is going to compare. So those groups are independent. Independent. They are not dependent on each other. They are not correlated with each other. Both are different groups. The results they are going to compare. Example I'm telling you. Two groups with high cholesterol participate. Two groups are there. They are having high cholesterol. The first group receives a new drug. The second group receives a placebo. Some other placebo, right? Then the experimenter means those person who are doing the experiment, they will hypothesize that after 2 months, your LDL cholesterol levels will be lower for first group than for the second group. So after 2 months, they will observe whether the lower of the means the what is that called? The LDL level, means cholesterol levels will be lower of for the first group. For first group it will be less in comparison to the second group, because second group they are giving the placebo. First group they are giving the drug, right? So in comparison to second group, the first group will be having little cholesterol after 2 months. Why? Because they are under treatment. So the result after 2 months, whatever they will get when they will compare, the first group will be having less LDL cholesterol in comparison to the second group. Why? Because first group is going to get the drug. And when they are going to get the drug, obviously they are under treatment. So obviously the cholesterol level will be reduced. But the placebo, they are not going to get any drug. So that is why the cholesterol level will be remain same, or almost, means it will be more than the first group. This is the thing I want to tell you, right? And both are independent, as I said, because first group I have given the drug, second group I have applied your placebo, right? And for two groups, T test, ANOVA, that is F test, are also interchangeable. Means we can interchange T test, either we can interchange ANOVA also we can interchange. All these things we can. Then coming to two matched sample t-tests. This is a test hypothesis that sample means are equal when observations are correct, correlated. Pre or post test data from match control. Example, I'm telling you, 30 singers completed singing voice handicap index pre and post treatment, right? And the mean score pre-treatment is 42.49, was significantly less than the mean score post-treatment, 27.5. So this is the comparison they have done between the pre and post-treatment. 30 singers they have taken singing voice handicap, right? So you have to test it whether it is correct or not. You have to go choose those singers and you have to go the test. You have to do the test on their voice. And pre-treatment they have got 42.49 and after treatment they have got 27.5. So after treatment it has been reduced the index, right? So this is correlated because singers are same in the first two and second same, but pre-treatment and post-treatment, right? And you can see here over 110 versus 210, you can see sampling distributions, regions of likely and unlikely values. Two-tailed test, you can see here, likely values, unlikely values. These are unlikely values you can see here. With this, we have demarked. And you can see here one-tailed test. This is your one-tailed test. So these are coming under those. Then when we are comparing the group means two groups are there. Suppose two groups are there. Each group will be having some, you can say the, okay. Okay. So comparing group means that is ANOVA, you can see one-way ANOVA is there, two-way ANOVA is there. One-way ANOVA used when there are more than two levels of single independent variable. When we'll be having two levels of independent variable will be there. Then test the hypothesis that all sample means are equal. So here what you have to keep your hypothesis, there is no significant difference, right? Test the hypothesis that all the sample means are equal. And statistics, if test also you can implement. So you can see here one way ANOVA. Features here recording of voice, music, or control sounds. Measure of change in heart rate. Mean changes, suppose music 4.68, voice 3.54, and control 2.18. The if test was significant, indicating that at least one contrast is removed. So this is a one-way ANOVA. One example I have given. Multi-way ANOVA, like two-way ANOVA, three-way ANOVA. And these are two or more independent variables will be there. But as you have seen here, only means between the groups we are comparing. But in this second class multi-way ANOVA, here multiple independent variables will be there. Two or more independent variables will be there, right? So two ANOVA, you can see here some of the experiment we have just showing you. Zhuang et al. 2009. Speakers with polyps, nodules, or nothings. Speakers, those are having polyps, nodules, or nothings. And you can see here sex, depending on sex and diagnosis. Two measures of airflow, that is PTF separate analysis. Result what they have got, significant effect of sex and diagnosis. Two dependent, you can see here, we have taken into consideration these characteristics factor sex as well as diagnosis. We are comparing. Then you can see here for PTF, post hoc comparisons. This one also, MFR. So many things are coming under your two-way ANOVA. Post hoc test, as I have discussed there. A significant if statistics means only that one of the group means is reliably different from other. Many out of many groups, one group is different from others. This is what it is showing about the post hoc test. Post hoc test actually it is identifying which specific contrasts are significant, which groups different from reliable. Means from others group, which group is reliably different, that you need to find out. Normally via a series of pair-wise comparisons. So what you have to do? Individually between the pairs, you need to compare and you need to check whether what group is different from others, the results. That is what coming under your post hoc test. Okay, how we are carrying out this post hoc test. One example I have given you here. If there are three groups being compared at the 0.05 level, that means 95% confidence level, each must be significant of the significant value should be 0.017. If not, it is rejected. If there are four groups, suppose 6 comparisons will be there, right? Comparisons, you can see significant parameter that is 0.0083 for each. And post hoc test, as you can see here, many kinds of post hoc test exist for comparing all possible pairs of names, like your skips, Turkish HSD, Newman Kules are most common, right? Then example of post hoc test, as you can see here, Newman Kules post hoc comparison showed that fetal heart rate, responses to music, voice didn't differ. So these coming under your post hoc test and repeated measures of designs, as you can see here, matched pair t-test are in simple case. Repeated measures ANOVA is more powerful technique. Okay, so these are the things you need to know whenever you are going to implement these in your pharmaceutical research. Overall, I have told you like ANOVA, t-test, chi-square test, pair t-test, right? So these are you are going to F-test, those you are going to implement. So repeated measures of ANOVA, as you can see here, mean and standard deviations, vowel, phrase, sentence, and counting. In some means English studies, not only the significant means your statistics you can apply to pharmaceutical research, any kind of research, whenever you are comparing some data, right? Multiple data you are getting after doing the experiment. Between the data, when you are comparing the results, then you can implement this one, right? So this is about your this one. And correlation regressions, as you can see here, correlation measures the extent to which knowing the value of one variable lets you predict the value of another. When one value will be useful for you to predict the value of another, that is called correlation. Means what is the correlation between that? And generally Pearson's parametric versions we are doing and non-parametric Spearman's, right? Pearson's you are going for parametric versions. Non-parametric versions we are going to use the Spearman's correlations coefficient, right? So I'm not, as I told you in the beginning, I'm not going to in details, right? Just I'll tell you what are the different statistical methods you are going to implement in research, right? Some of the example I have shown you here, right? And in the regression, you can see here, regression is basically the same thing as correlation, but only the exception, you get an equation that lets you predict the exact value. Here you are going to predict the exact value. There through by using one value, you are predicting another. But here you are predicting the exact values rather than just a measure of how good that prediction is, right? So this is about your regression. Okay. And example here I have given you which acoustic variables are most strongly associated with the perception of model versus falsetto voice quality, right? And coming to correlation and regression limitations, what are those? R square versus significance, the N issue. These are the major challenges when you are implementing the correlation and regression. Okay. Then coming to use of this statistical method in pharmaceutical research, let me tell you, introduction of pharmaceutical industry, drug development process and clinical trials, biostatistics in clinical trials, as I told you, because there you are going to design a study, you are going to select the patients, number of patients will be high depending upon the Clinical trial, you are going to do clinical phase 1, 2, or 3, whatever. Study design, clinical data, statistical modeling, other topics, right? And coming to the drug, as you know, the definition of drug, this is a drug, this is substance either from chemical origin or any non-synthetic origin, which is used for mitigation of disease, right? So different types of drugs are there. Means branded drug, you are going to develop branded, and you are going to go for a generic drug. Okay, that is not of our major concerns. And in pharmaceutical industries, as you can see here, list of 2013 top biotech and pharmaceutical companies: Johnson and Johnson's, Novartis, Roche, Pfizer, Sanofi, all this. Okay. And coming to list of 2013 top biotech and pharmaceutical companies, as you can see here, Pfizer, Johnson. Okay. Later on, it is changed depending upon their outputs. Okay. So you can see here the changes. Okay, sorry for the interruptions. Actually, yeah, I am in a position that I have to handle so many things. That is why last time also my program was cancelled. So sorry for the inconvenience. And today also many interruptions are there, but still I'll continue with forgiveness of you. So pharmaceutical industry, as you can see here, later on change depending upon their outputs, out results. And you can see here pharmaceutical industry in R&D about number of new drugs, one. 10 years of R&D spending, $13.2 billion, as you can see here. Sanofi, 6 drugs. For that, they are spending 60. AstraZeneca, 38.2 for 4 drugs. And Pfizer, for 10 drugs development, they are spending around 77.8. So you can see here, Pfizer, they are spending more amount for development of more number of drugs. Also, you can see here, Johnson and Johnson, for involvement for evolution of 13 drugs, $67.6 billion, right? This is the CRO, contract research organizations, how the pharmaceutical development is going on, right? And in this, you can see here what are the study design. As I told you, what is the statistical model we are going to implement. Here, drug discovery, different phases are there, right? So for the drug discovery, it will take 3 to 6 years. Preclinical, including the preclinical level. Preclinical level generally it is done on animals. Then coming to clinical trials, it will take around 6 to 7 years. Just you check it. 7 plus 6, 13 years. And here another 2 years. So for getting FDA approval of one drug, it is almost taking 15 years. So how much difficult it is, how much difficult task it is. And in the clinical trials, as you can see, phase 1, phase 2, phase 3 phases. Phase 1, they are taking 20 to 100 patients. Phase 2, they are taking 100 to 500 patients. Phase 3, they are taking 1,000 to 5,000 patients. And on that, they are going to implement the result, means the statistical models, and the results they are obtaining, they are comparing, right? And depending upon the results, they are sending it for the FDA approval. Okay. Then clinical trials. In the clinical trials, that is your, as I already said, no need to discuss about that. Clinical trials means the drug will be tested on your human beings, right? And phase 1, I don't want to talk much about this. Phase 1, phase 2, phase 3, what is that actually? These are clinical trials basically done on your human beings to check the toxicity of the drug, right? And phase one is done on volunteers, healthy volunteers. Phase two, phase three, phase four, they are done on your patients, right? And no need to discuss all this. Conduct of clinical trials, as you can see, study planning, study startup, study conduct, then study course close out, then analysis and reporting. There you are analyzing and reporting, right? Agenda, you can see biostatistics in clinical trials, study design, clinical data, statistical model, right? Okay, design of study and develop the protocol. Design the randomization algorithm if needed, right? Biostatistics, as you can see here, study classifications, phase 1 PK, PD studies, through QT studies, superiority studies, dose response studies, all these they are doing in biostatistics. Okay. And as I told you, they are going to implement all these designs, conventional study design, other study design, like parallel group design, crossover design, titration design, cluster randomized design, group sequential design, all this, as you can see, a study design. Patients run in randomization, then test and control A and control B, different group. Then they are comparing the data. So like this, another one, as you can see here, patients randomization like in your bioequivalence studies, right? Sequence and sequence 2 test they are applying, and this is your washout period. After that, phase 2 means the period 2. In the period 1, test and control. After that, washout period. Washout means the elimination phase through urine or fecal matters. Whenever you are giving some drug, after that again you are comparing the data. So like this, we are applying the biostatistics in clinical trials for the drug development. So like this, so many examples I have given here, and as I said you, this is the significance of biostatistics in pharmaceutical research. We can implement anywhere, any disease like diabetes, stroke responses, cardiovascular endpoint study, pharmacokinetic study, right? So everywhere we can apply the statistics and we can compare the results. In diabetes, as you can see here, running period 4 weeks, it will be an insulin, glargine, metformin, NPH metformin on this, right? Case 1 studies, this is one. So like this, we have done some of the things. Just some example I have shown how we are implementing this particular biostatistics, means statistics in pharmaceutical research. Okay, like cardiovascular disease, we have chosen placebo, and some patients we have given atorvastatin drug, and we are comparing the data, right? And like other things, pharmacokinetic, we are checking their means rate of elimination, Cmax, Tmax, your plasma concentrations, volume of distributions, all these were compared, right? So this is what I want to say, the implementation of your biostatistics or statistics in pharmaceutical research. So current statistical research topics, as you can see here, adaptive design, missing data in imputations, dose finding, randomizations, risk-based monitoring and fraud detection, design and analysis of non-inferiority studies, mid-pace biostatistics, right? So like this one. So this is about means I have shown how we are implementing all these means by statistical methods into pharmaceutical research, and I hope the talk is clear to all of you, because this is a small time period. Within that stipulated time, I need to finish. That is why I have gone fast. And if you need these particular materials, then you can write to research circle and I can circulate the materials. Thank you. If any questions will be there, please.
Thank you so much. Thank you so much, sir, for this very informative session. If anyone has any question, you can ask. Thank you so much, all participants, for your active participation. Let me inform you to all that this complete session will be available on our Research Circle YouTube channel. So if you want to watch it again and want to share it to somebody, please do visit the Research Circle YouTube channel. Thank you. And last but not the least, thank you, Dr. Nalini Kanta, Sahu for taking out the time for session. With your permission, I would like to end today's session here. Thank you, sir.
Thank you. Thank you very much, madam. Thank you.